thank you very much for accepting this invitation for Moving Along newsletter. It's a huge pleasure to have you here with us today, Niccolo. So in this issue, we are focusing and celebrating the MDS Congress in Madrid that took place a few weeks back. And in general, I think that the first thing that come to my mind is that we were all so happy to meet in person finally. So I wonder, what do you think? Are you preferring online meetings or in-person meetings?
I mean, this is a question that, of course, I was so happy to be back in person. I'm kind of affected by Zoom fatigue, so it was so refreshing to be back in person. It felt amazing. On the other hand, I think we were not used anymore to so much social interactions.
Yeah, it was actually overwhelming.
It was a little overwhelming, but fantastic overall. Fantastic overall.
And
yes, absolutely.
Talking about fantastic, congratulations on your tremendous lecture. I really enjoyed it. And I think that it was very, very difficult to choose research for your highlights of basic science in other movement disorders session. So how did you approach to that?
Yes. Well, first of all, thank you very much. I really appreciate your kind words. And yes, it was actually quite a bit of work. When I first got an invitation, I was, of course, incredibly excited. Then I realized what task I was asking and say, oh gosh, this is going to be a lot of work. And indeed it was. Indeed it was. So I used different approaches. I first tried with a systematic approach. So disease by disease, I tried to see all the most prominent journals spanning from Science, Nature, all the journals of the Nature group, neuron cells, movement disorders, PNAS, all the biggest journals, what they had published in the last year regarding the different movement disorders that I was interested in, and that covered Huntington's, dystonia, myoclonus ataxia, and tremors. And so this is how I screened initially what relevant papers had been published, and I created a base of papers that I thought I would be interested. And then my second step was, among the papers that had been published, what stories were the most new and potentially most interesting. And I ended up also, after talking to some persons that advised me to select only a few topics that I thought would be of interest, rather than covering the entire publication, all the publications of the last year, which could have been overwhelming also because these were basic science works. So to go into details could have been overwhelming for you. I chose a couple of stories that could be particularly relevant, either for translational purposes or because they were talking about something particularly new, potentially controversial. And so, yeah.
This is very. This was very good idea in the end. So you choose two diseases in general: Huntington's and dystonia. Yes. So in Huntington's, it was very interesting to know that in general it is said that the longer CAG repeats are associated with earlier onset of motor symptoms in Huntington. But you said that it's only true in 50% of people with Huntington. So what's new?
Yeah, so actually, this has been known for a while, and there have been a number of genome-wide association studies that have been trying to discover what else is driving age at onset and clinical progression in patients with Huntington disease. And this is both to help with understanding and predicting when someone with a defined expansion will develop disease and understands the clinical progression, but also for obvious therapeutic translational aspects, because if you understand what slows down disease onset or disease progression, that's something that could be targeted therapeutically. And there have been a number of very wide and comprehensive genome-wide association studies that have identified genetic loci associated with either an earlier or later age onset than what predicted based solely on the expansions. But nevertheless, these genome-wide association studies, first of all, they're not explaining everything about what else is going on genetically in terms of what's determining the age at onset. But also, if you think about genome-wide association studies, these are always single nucleotide polymorphism that are located in non-coding portions of the genome. So sometimes it's hard to go from an association to what it's actually going on at the biological level. So the article that I chose, I thought that was very interesting because it's the first study that's linking coding variants in a specific gene. In that case was FAN1, FAN1, was linking coding variants, so they are altering the function of the protein. And the authors of that study actually performed very elegant studies that were actually showing how mutations that affect the function of the enzyme were actually maintaining the stability of the CAG expansion in cell lines derived from the patient. So really going from genetic association to an actual biological mechanism, very, very elegant study. So
the study that you're referring to is the McAllister study, right? That used those big cohorts of Predict PD and Register PD.
Yes, exactly. So they perform whole exome sequencing study in patients with extreme phenotypes, either extreme in the sense of more severe earlier onset or vice versa, less severe later onset than expected based on CAG expansion. And then they do burden gene analysis to see whether there are genes that are enriched for rare damaging variants present in exome sequencing data associated with either an earlier or a later age at onset. And turns out the FAN1 was, with the analysis they performed, the only gene that passed exome-wide significance, so there was significantly associated with a more aggressive or a more benign phenotype than expected. And then they perform functional characterization to see how the activity of this enzyme is actually controlling genomic stability of the CAG expansion.
So is it true to say that this FAN variant can be used to predict the age of onset in Huntington's?
Well, let's say that it's definitely true. And we can say that if someone has a FAN1 variant that's affecting the enzymatic activity, the cleavage activity of the enzyme, the onset is likely to be anticipated, and the disease more likely to be more aggressive than what expected based on the CAG repeat detected in the blood. Because what the field has been discovering is that CAG repeat expansion, they're not a static entity, but they keep expanding, and that's what's driving pathology.
And do you think that it can be also a target for any therapeutic approach?
Absolutely. This has been already developed. That's true for FAN1, but also for other genes that have been linked through genome-wide association studies to Huntington age at onset, MSH3, MSH1. Absolutely, this is a very promising areas for new treatment development. Absolutely.
I was wondering whether FEN1 is it only only a factor to predict the onset of Huntington, whether it will be earlier and the course will be more aggressive, or is it also linked with the motor symptoms or non-motor symptoms in Huntington? Was it studied before?
Yeah, it was addressed as well. They were showing that there was more severe motor progression and more severe progression to cognitive decline in carriers of these damaging FEN1 variants.
Okay.
But it was also very interesting to see that some other variants located in a different portion of the protein were associated with the milder. So each variant may have a different effect, but definitely a critical gene and protein for affecting the size of the CAG and the somatic instability that's driving disease progression and the progression of pathology. Yeah.
Okay, that's great. And then you mentioned, or your second part of your lecture was on dystonia, and you mentioned that this is your favorite disease, if I can say that. So first of all, I want to know why this is why dystonia is in your interest.
Well, it's because I trained with Kailash Bhatia, and Kailash is someone who inspires very strong passion in people working with him. So he taught me about dystonia. I knew nothing about dystonia before working with Professor Bhatia, and I started working with some of his interesting families, and he's taught me about dystonia, and I started working on identifying genes for dystonia. And then from there, I started thinking about how dystonia develops, what are the mechanisms of dystonia. It's a very complex condition which probably groups together diseases that present clinically with dystonia, but from a biological point of view, they are very different, and genetics can help to put some order in what are the different dystonias. And
in your lecture, you focused on type 1 DYT6 and slightly on DYT1. So those DYTYs are your also your favorite ones, or you found it very interesting in terms of basic science?
Well, I thought that those were the highest quality publications.
Yes.
And interestingly, there was a clear story to tell about mechanisms of the YT6, TAP1 related dystonia, which, so there were several papers that were basically going in the same direction, which is something somewhat unexpected, which was suggesting that one of the basic mechanisms leading to TAP1 related dystonia has actually not to do with the role of TAP1 in neurons, but more to do with the role of TAP1 in the development of oligodendrocytes, which are essential for synthesis of myelin. So a paper from
Domingos, right?
Domingos et al., yes, published in the American Journal of Human Genetics, that did a tremendous work in reprogramming iPSC cells from patients with different genetic mutations causing TAP1 and correcting them with CRISPR. And then they do very extensive transcriptomic analysis looking at the shared pattern of transcriptomic dysregulation. And the most prominent dysregulated pathway was oligodendrocyte maturation and genes involved in this function, which was very interesting. Then and that linked to another part of my talk, which mentioned some of the work performed by Dr. Bildauer. Two important papers, one showing that in a mouse model where they try to understand the mechanism of a specific TAP1 mutation linked to dystonia, they find that this mutation causes dystonia by not recruiting this transcription factor YY1. And then, well, I've actually knew quite a bit about YY1 because my group published a case report reporting the first case with dystonia with YY1. So this YY1 transcription factor, which interacts with TAP1, is a new cause for dystonia. And the function of this YY1 is regulating maturation of oligodendrocytes. So I thought that there was a very interesting story there about the interaction of TAP1 and YY1. And then again,
sorry, can you tell us more about the phenotype of those patients with YY1?
Yeah, it's actually, so there are now 3 or 4 cases. I think it's 3 reported in the literature. Now, what is interesting is that from a movement disorder point of view, they have a presentation that it's really close to that of patients with TAP1 dystonia. So from generalized dystonia, but with prominent oromandibular craniocervical involvement, more than lower body involvement as seen in the YT1 dystonia. And interestingly, this gene was initially discovered to be linked to a form of a syndromic intellectual disability called Gabriele de Vries syndrome. And interestingly, these patients, a couple of them had some degree of intellectual disability, but one was reported to be normal from that point of view. So the movement disorder was truly the most striking aspect. So dystonia with prominent oromandibular involvement in all the cases reported.
Okay, and then you also mentioned about the lysosomal dysfunction.
Which is something,
And you asked the question, how does it fit to dystonia? So how does it fit to dystonia, this lysosomal dysfunction?
Well, the truth is, we still don't know, but the paper I discussed suggests something that I've been thinking quite a bit about this because I'm in a lab where lysosomal dysfunction is studied in the context of Parkinson's disease. And I've been very excited, and that's something that actually my research is involved in trying to understand how certain genes that are linked to lysosomal dysfunction, that are involved in lysosomal function, have now been linked to dystonia. And I thought that, again, a paper from Bill Dower's group was very interesting in the sense that was proposing that in a mouse model of TAP1 related dystonia, they were finding that there is a defect in glucosaminoglycan catabolism. So these glucosaminoglycans were accumulating, and they found that TAP1 is actually involved in regulating the expression of a lysosomal gene. And this lysosomal gene is involved in degradation of these glucosaminoglycans. And accumulation of glucosaminoglycans when this lysosomal gene does not work may lead to defect of oligodendrocyte precursors maturation, which is starting to become a convergent theme, at least for TAP1 dystonia. But that led me to the other story, which I thought was very interesting. VPS16, VPS41, VPS11 are genes that are forming, protein product forms a protein complex that's called HOPS, which is essential for maturation of lysosomes and for autophagy. And they are now all being linked to dystonia. But a paper that emerged that came out last year is that there are patients that have homozygous mutations in VPS16, and that they don't have dystonia, but they have a lysosomal storage disorder with leukodystrophy and with mucopolysaccharidosis with accumulation of glucosaminoglycan product. Overall, suggesting that maybe that's a potential explanation for why certain genes involved in glucosaminoglycan metabolism, hence regulating oligodendrocyte maturation, may link lysosomal dysfunction to dystonia pathogenesis. But that's just one potential mechanism, but I thought that all these papers were like fitting very well in indicating this.
Linking. to each other. So the last thing will be ritonavir. So you mentioned about that, and so what do you think? Is it a kind of a breakthrough in the field of dystonia, or?
Well, I don't know, but as certainly great potential. The work is very elegant and conducted by the group of Dr. Kalakos, Duke University in North Carolina. And
it's based on animal models, right?
Yes. Well, they do two models. They first look in a cell model of DYT1 related dystonia, Torsin 1A related dystonia, where they do a drug screen, trying to see if a battery of FDA approved drugs, I don't remember the exact number, but it's a few thousand FDA approved drugs, they can modify a cellular phenotype of Torsin 1A related dystonia, which is protein aggregation of the mutant Torsin protein. And they, amongst the most effective drugs that were able to rescue this phenotype, there was ritonavir, and they which is a protease inhibitor that's widely used for treatment of HIV infection. And then they go on and study the effect of ritonavir in a mouse model of the DYT1 dystonia. As you may know, unfortunately, this mice model, they don't have a dystonic phenotype, so that's a potential limitation. But her group elegantly proves the several developmental phenotypes that are seen in this mouse model of the DYT1 dystonia that were fully corrected, including one of the most widely accepted neurophysiological phenotypes of the DYT1 dystonia, which is the paradoxical excitation of cholinergic neurons in response to dopamine stimulation. And so an important thing, and I had a chance before my presentation to talk to Dr. Kalakos, and she mentioned the doses that were able to rescue these phenotypes are those that are reached, that are used therapeutically in patients with HIV infection. So this would be doses that would not cause toxicity. What we don't know is if they would be effective in passing the blood-brain barrier and if the pharmacokinetic would work. So there are many questions to be asked, but it's definitely, from a mechanism point of view, very interesting because it seems that this is working through the modulation of this pathway, the integrated stress response pathway, which is again in a converging area that may be important for different forms of dystonia, so not just the DYT1.
Yeah, that's really, really interesting. So let's keep fingers crossed for further studies, right?
Yes, absolutely.
So, this would be it in terms of your highlights during the MDS session. For this interview, I would like to ask you also about your current research. So what is your research now?
Yeah, so I've three main areas of my own research. One is purely someone was calling me. Mostly genetics. So I keep working in my group about identification of new genes linked to dystonia, and I also, through the GP2 consortium, Global Parkinson Disease Genetic Consortium, trying to identify new genes linked to Parkinson's disease. So still very active in this. At the same time, I have two projects that I'm carrying out that are more biology focused, understanding the mechanisms of how lysosomal dysfunction may contribute to dystonia. So, and that's using iPS-derived neurons from patients that have mutations in genes linked to lysosomal dysfunction and dystonia. So that's a big area of research, and that's why I was so excited to present that data about lysosomal genes and dystonia. And then I'm studying a mouse model of a gene that I published together with Professor Bhatia, PD10A related movement disorder. So I'm trying to characterize in a mouse model how PD10A mutations cause disease, and understanding how this interesting molecule could be targeted therapeutically for patients with different movement disorders. So different projects. I keep myself busy.
Yeah, I can see that. There are very interesting projects, and I wish you all the best. So thank you very much for your time and for this great interview. I learned a lot, and I hope that there will be a huge interest to see that to see this interview, because it is very nice to hear your enthusiasm on your research, and I really enjoyed it. So thank you very much.
Thank you, Kaza.